This one we're going to study how UV light can act as a mutagen, meaning that it can cause mutations. UV light is a known mutagen. It's known to cause damage to the DNA. That's how it causes the mutations. We're going to work with Serratia marcescens. Serratia marcescens, when grown at room temperature, has a red pigment to it. And so what we're going to do is expose the bacteria to UV light for different time periods. And what we're going to see is if that exposure to the UV light will cause a mutation where it will lose this red pigmentation. So we're going to grow all of the plates at room temperature. So under normal conditions, it should be red. But what we're going to do is we'll have our control, which is not exposed to any UV light, and then we're going to expose the other sample plates to different time segments or exposure times to the UV light. If the exposure time is relatively short, then typically you would not see a mutation. It is too short to cause that damage to the DNA, which results in a mutation. But if the exposure time is too long, actually it can end up killing the cells. So the timing of the exposure time is very critical. So we're going to try to see what time will cause the mutation and have a change in that pigmentation, but not kill the bacteria. Excuse me. So before the UV light exposure, the control, at room temperature, this is the plate with the complete lawn on here of Serratia marcescens. You can see it has that reddish pink color to it. This is an example after 30 seconds of exposure to UV light where at this point there is no growth. It has killed it. We went for different time periods from 5 seconds, 10 seconds, 15 seconds, 30 seconds. And so this is showing the two extremes. Like I said, the timing is critical with UV light. Very brief, short amount of time is not going to cause a problem. Too long of a time will kill the cells because it causes damage, at times it will actually result in death of the cell. We use that sometimes for sterilization purposes, like light goggles that we use. We put them in a case and we use UV light to sterilize them. Labs sometimes at night have special UV lights that they will turn on or activate after the last person leaves to it'll be effective in cleaning the tabletop or exposed surfaces, the air. But as I said, there's that timing where in the middle, between not causing damage and death, where it can cause mutations. So this is an experiment where we test that theory out.